Morning, everyone. I hope everyone's fine. We'll just wait another minute so that others can join in. We see a lot of attendees joining at the moment. I'll start in exactly 1 minute. In the meantime, feel free to go through the handouts if you can. If you have any questions, feel free to post it on the chat. I'll go through the housekeeping rules in 1 minute. All right. Let's start these. So welcome to this refresher training courses. So this week we'll be focusing on the nucleic acid purification as well as the next generation sequencing, next portfolio. Before we start, I would like to have some housekeeping rules. Feel free to post your questions in the chat box. If you can see the chat box, please feel free to post your questions or comments you have anything during the session. The questions and answers will be addressed at the end of the presentations if it is okay, but feel free to chat. Okay, so just as I mentioned in the beginning, this is just day one of the refresher training. Just want to remind you at the same time tomorrow we have the continuation of the nucleic acid from nucleic acid chemagen series. Feel free to please join it. We have also added some brochures. Please download it, and if you have any questions, feel free to mention it in the chat. Anything you feel like you're having some technical issues joining, if you have any, please reach out to the team and we are happy to help you out. With that, I would like to introduce today's speaker. So today's presenter will be Dr. Catherine Go. Dr. Catherine Go is the product manager at Revati, specializing in automated nucleic acid extraction instruments and kits. Prior to joining Revati, she holds a PhD in Duke and US Medical School in Singapore, and also has a master's in pharmacology. She is a Marie Curie postdoctoral fellow and has strong foundational background in RNA biology. So prior to joining Revati, she gained a valuable experience in product management and drug discovery, and her main focus is also on the nucleic acid-based technologies. So welcome everyone, and I give the desk to Catherine to her. Thank you.
Thank you, Philip, and thank you everyone for joining. And thank you for the kind introduction, Philip. So I hope you can see my slides all right. I'm just going to move on. So I'm going to be talking about the nucleic acid purification solutions from Revati, which is under the chemagen brand name. And just to give you a background about chemagen, we were initially founded as a German company near to Aachen. And in 1997, they were founded and based on the proprietary technology of magnetic polyvinyl alcohol beads. And since 25 years, they've been producing these beads and also the separation technology for manipulating nucleic acids. In 2010, they were bought by PerkinElmer, and that also enabled them to launch their products in global markets. And just 2 years ago, we then had a name change to Revati, where we focus on life science and diagnostics. So our solutions are installed in labs, diagnostic labs or biobanks or research labs that are doing high throughput automated nucleic acid extraction. And we have over 25 years experience in this area. Our technology is really known for being best in class in terms of nucleic acid integrity, and also a very versatile solution. So I'll go through with you in more detail here. The founder is Dr. Lothar Prasad, and he still is managing the chemagen site in Basweiler. And I think as he mentions here, we always place customer first, and that really drives our product development. And as you know, we're focused on automation, automating the nucleic acid purification. And this is something we want to stress as well to customers, because there are many advantages of automating nucleic acid extraction. And I think there is a movement, as you can see, with a lot of genetic testing being done, more drive into personalized medicine, and the need for really standardizing the quality of nucleic acids that you obtain for your molecular assays. So we want to focus on selling these automated solutions. We have manual kits as well, but they're not our focus. So really the focus should be on getting labs to automate their nucleic acid workflows, and the key arguments are here. So going into the chemagen technology, it's based on two core pillars. Well, one is the MPVA magnetic beads. So unlike commercial beads, which you see on the bottom right, I put on laser, you see here, most of the conventional magnetic beads would have a core surrounded by paramagnetic particles, in turn coated by polymer coating, whereas the MPVA beads from chemagen, they consist of a polyvinyl alcohol that has been cross-linked, and you see that captures the magnetite particles in position. And the advantage is that you can pack a lot more magnetite in each bead, so they're moving faster, even from in large volumes. And the matrix is also very robust. It can be put through different liquid handling processes. It's shipped at room temperature. It's even autoclavable. You can also functionalize the PVA groups on the beads with oligo-DT or streptavidin to be able to bind to different targets. And lastly, the surface of the bead is hydrophilic, which results in less interactions with proteins, so you get a higher purity of nucleic acid at the end. So the other core pillar of the technology is the separation technology. And on the left here, you see the chemagic 360 instrument. One thing to note, which is the real advantage here, is the interchangeable rod head system. So you can take out this rod head and switch between different formats of 12 rods, 24, or 96 rods. And this not only enables you to manage your throughput, it also produces a large sample volume range. So we have the market's, I think, the widest sample volume range capability from 50 microliters up to 18 milliliters. These rods you see will go through this electromagnet, which is a switchable, enabling the magnet to be switchable. So when the current is on, the rods get magnetized and they go down into the process solutions. I have a better view of it here. And you can see how it works as well. So the rods will go into the either deep well plates if you're doing a 24 or 96 well reaction, or 50 mil tubes if you're doing 12 well reactions. So you can see that the rods are going into the lysis solution and they're rotating. And this is another key aspect of the instrument, the separation technology. The rotation resuspends beads, allows it to interact with the nucleic acids, and then the rods move the beads into the different wash buffers and then elution buffer. And this rotation is very gentle. We've compared it to other automated platforms which also may use rods. These tend to go up and down, or other platforms that use pipetting, and these tend to shear the DNA or RNA. So in terms of the integrity of nucleic acids, you can achieve a very high molecular weight of DNA with the chemagic separation technology, suitable even for long read sequencing. Another key argument is the dispenser. So you see this at the back here. This is connected to your lysis buffers, binding buffers, wash buffers, and so on. And this enables automated dispensing of your buffers. So it's a semi-automated system, the chemagen 360. You only have to load your sample, the beads, and the elution buffer. And all the extraction also occurs quite quickly with about an hour processing time. So here are the benefits. As you can see, the different rod head sets. This really allows a range of sample volumes. Obviously, with the lower throughput rod head set, it's more dedicated for higher volumes. But even with the 24 rod head, yeah, you can go down to 200 microliters and up to 10 mils. And with the 96 rod head, that allows you a higher throughput and even up to 1.5 mils for plasma and 800 microliters for blood. So this is a very versatile instrument, which many labs appreciate to be able to put in different workflows and extract different sample materials on the same instrument. So as mentioned, we have a lot of experience, largely in human sample material. These are the most common types of human sample material we deal with. It's not limited to this. We also have now experience in wastewater processing, some soil, also cultures, E. coli cultures, and so on. But a lot of our customers are doing largely blood or buffy coat, saliva, and these kinds of sample material. And we have various kits to allow the isolation of different targets. There are some universal kits. In fact, our blood kits can be used to extract human genomic DNA from blood, buffy coat, and saliva. So there are different sample materials you can analyze with one kit. For different targets, though, like RNA and cfDNA, we have dedicated kits for those. So our chemagic brochure you see in our attachment would give you an overview of what kits we have. The other benefit that using magnetic beads with these chemagic separation technology brings is the low risk of cross-contamination because you're moving beads and not liquids. We've spiked a high amount of HSV virus here in the blue wells with water in the neighboring wells, and you see that when you perform the whole extraction, followed by a qPCR, you don't see any viruses in the neighboring wells. So there's low risk of cross-contamination. The chemagic 360 also employs the use of a drop cover with the larger volumes. So, and our extraction instruments are used in diagnostic labs with IVD kits available. So a lot of the extraction has undergone a high QC. With regards to purity of nucleic acids, we also meet the required purity ratios, which is about 1.8 to 2 for 260 to 80 and 2 to 2.2 to 260 to 230. You can see from this graph, which was provided by a customer, Life and Brain, that even for blood stored at 3 days, you are still getting consistent extraction results. And even with free store cycles of 2, you still can get good quality DNA. And they have used the chemagic 360 extracted samples, and the DNA extracted 16 years ago are still being used as an internal control. So it's the quality of DNA you're getting is very stable. Yeah, this also is due to the fact that we use different reaction vials for each step. As you saw the turntable, each step contains a different binding and wash buffer, so you're avoiding the carryover of inhibitors. This is the key argument, as I was saying, with our high molecular weight DNA. This is a femtopulse plot, which can have the resolution for long fragments. And you can see that from extraction of fresh blood, we are able to get DNA fragments, majority being in the 100 to 200 kb. lengths, and this is really unique to our technology. We've done a side-by-side comparison with other brands like Kingfisher, for example, from thermal, which is used in different brands and different platforms. And these tend to give rise to fragments of about 40 kb and below. So this really attests to the unique advantage of using the chemagic separation technology, and is appreciated by biobanks that do not know the downstream assay. So need to start out with a high quality of DNA that can be used in different assays. And even for human genetic testing labs that may be employing more long read sequencing, this is definitely a key feature that you should highlight. So again, this is due to our gentle stirring resuspension, and the fact that we don't use any heating as well in our extraction. So these are the different sample materials we deal with, and we also get very high and consistent yields. It's good to know the theoretical yield you can get out of blood. So this is typically between 20 to 50 micrograms of DNA per mil of blood. So when customers are asking you for if this can give you 100 micrograms, or you have to also realize what is theoretically possible. And I think 50 micrograms per mil of blood is really the maximum you can get, and most technologies would produce yields about this range. And the nice thing about chemagic technology, it's really robust. You can analyze a lot of samples on the platform, and there are very few dropouts or failures, and you get very usually tight, consistent DNA yields. And we've used our instrumentation for large population studies, looking at hundreds and thousands of samples, and this is really, an advantage that users like. So with different kinds of material, you can expect different yields. For saliva, for example, it might be more variable. But yes, we have experience in many different sample types. So apart from the high purity, yields, and integrity of the nucleic acids, we can also cater to the large flexible sample volume ranges, the different extraction targets and types of samples, and fast runtimes. And what's more, the chemagen team, they're still based in Basweiler. It's where the kits are produced. So we're really owning the full value chain in terms of manufacturing of the kits. So if customers need a dedicated kit with a custom buffer or a different amount of a certain component, this is something we can cater to, which is also an advantage. It's not something that other companies can so easily do. We also sell our kits in bulk formats, which allows it to be more cost efficient when labs are running high throughput extraction. And of course, everything undergoes a high level of QC with our labs being ISO certified. So just an overview of our chemagen products. These are the instruments in our portfolio. I've shown you the chemagic 360. We have this available both as RUO and IVD versions. It takes up about half a bench space. And we have also the chemagic Prime family. These are the fully automated solutions where we have liquid handling also from Revity integrated in an instrument. And this allows a full walk-away solution, preparing from primary sample tube even down to downstream concentration normalization or PCR setup can be used with this machine. We recommend sample volumes of up to 10 mil, so up to the 24 and 96 watt head here, just to cope with the liquid handling capabilities of the liquid handler. And this would take up a full bench, comes with a table. We also have a smaller brother of the chemagic Prime, the Prime Junior. This comes with 2 Pepitos on the right instead of a 360, as well as a Janus liquid handling with a 4-tip channel instead of an 8-tip channel. The Pepito is the benchtop instrument, very small and low throughput version of the chemagic system. It can process up to 12 samples, but it also uses the same extraction technology. So you have also the rotating rod heads, and you can it's carried out in tubes as well as a deep well plate. And the only thing to note is it can go up to 1 mil. It has the same size rod head as the 96, so the sample volumes here are limited to the same as the 96 rod head. So I would say here 800 microliters of blood and 1.5 mils of plasma, which is the same for the Prime Junior. But the nice thing to note is that all instruments rely on the same separation technology, so it's easy to scale up and down, which avoids validation efforts for labs and also allows you to, for example, get a foot in the door with the lower throughput machine and demonstrate performance advantages, and then be able to scale up to higher throughputs. So it allows also labs to be able to scale up in the future. Right, so I have a couple slides that will go through this, but I don't want to spend too much time. These are the main features of the chemagen 360, which I've mentioned before. You have also additional add-ons. You can see from our order guides, which you can get from Philip. So the ability to add a heating rack if needed, or UV light, or a switch box to link up two kits to the same instrument, to be able to switch easily between workflows. So this is really the most, I think, versatile instrument one can get in the most protocols we have for. The Pepito, as I mentioned, is a lower throughput instrument. The kits are sold as 6 packs and are hung on the side, and it also has the same extraction technology. And yes, this is the Prime, which I'll go through more in the second talk tomorrow, and the Prime Junior. One thing to note is that Revity also has automation solutions with the ability to implement even QC and with the lab chip and normalization with the Victor Nevo in these automated robotic workstations. So the chemagen 360 can essentially be integrated as multiple units hooked up to the same liquid handler to be able to really maximize throughput. And this was done during COVID days, where we could achieve like more than 10,000 samples per day. So if the customer has a need to really fully automate or have a high throughput and they want to automate even decapping and recapping, this is something that we can achieve through third-party vendors. Lastly, our kits, as I mentioned, they're manufactured by us. They're shipped at room temperature. That all the plastics are included in the kit, which is something you can't say for other vendors. Like Thermo, for example, sells plastics separately, which can add up to costs for the end user. For us, everything is included, deep well plates, the tips for the rods, and so on. So customer doesn't have to worry about this. We have CE IVD versions as well, and as I mentioned, they can be customized. And lastly, the beads we also sell separately. They can be functionalized with different groups, as you see here. And we have a OEM team that also allows these beads to be sold as in other brands, so as PCR kits and so on. So if you have questions or you have needs from your region with just needing the beads or even our buffers, just let us know as well. I'd like to go through as well our customers now and their workflows. And just to provide you a summary, I know it's a busy slide, but this is an overview of all our customer profiles. So the kinds of customers that are using our nucleic acid purification solutions. And you can see the main ones, I would say, are the genetic testing and the biobanking, as well as the microbiology customers. So these are largely for genetic testing, diagnostic labs or service labs that are dealing with a high number of samples. They are processing usually blood, buffy-coat saliva, or various different kinds of materials, usually extracting also diverse targets. You see also now a rise in cfDNA or microRNA. For them, they like the versatility of the instrument and also its consistent performance. And with biobanks, these are institutes that store samples, but sometimes also carries out extraction. As I mentioned, they like the ability that we have can achieve high molecular weight DNA. We see that it depends on the biobank, how much throughput they have. If they're involved in large population studies, however, this can be a lot. We also have core facilities. These are smaller groups, labs, and academic institutes usually, having also sequencing services and so on. These are lower throughput, but can be also um, a good customer to have. They're quite loyal, and they're also like our versatility and our consistent high yields. An interesting customer that you may not be aware of is the donor screening customer. So these are labs, for example, the Red Cross here in Germany, or stem cell donor registries like the DKMS. Uh, these are labs that have to screen blood donations or organ donations or stem cell donations. So they have to do HLA typing. Sometimes they're doing this with sequencing. They have to screen sometimes for parasites. They handle quite a lot of samples as well. So they're actually a really good customers of ours. We have a good footprint with them in Europe. They are doing mostly blood, saliva, buffy coat, and so on. And again, they appreciate the consistent performance. Cancer diagnostics lies in the umbrella under genetic testing, but you do see more and more labs that are focused, or companies and biotechs focus on cancer diagnostics or precision medicine. And there we have a nice ability with the chemagen 360 of extracting from large volumes, which you see being beneficial for being able to isolate low abundant targets such as cfDNA or circulating RNA. And with microbiology, this were our COVID customers, as you know, and infectious disease testing labs. So hospitals or clinics that are doing pathogen screening. They also like our technology because it's fast, it's versatile in the sense it can extract from different sample materials, and we are able to also meet their needs for any custom modifications. A very small but growing field of customers are the pharma, biotech, and CRO. These farmers and biotechs, they also may run their own clinical trials and perform their own extractions, or they may be doing more drugs research, so preclinical research, which involves plasmid isolation or RNA extraction from tissues or cells. So even IVT RNA, if they're doing manufacturing of mRNA therapeutics, for example, there have been requests of extracting in vitro transcribed RNA. So this is quite a nice a niche area which where we have not so much experience, but still we have some customers, and this could be something of interest. So I'd like to ask Pravik maybe to bring up the poll right now, which would ask you what kind of customers you see in your region that may fall within these categories. So it'd be great to hear what you think, um, what kind of customers you cater to mostly. Okay, so I'll keep going as that comes. So with the human genetic testing, we have different workflows. As you know, we have it kind of covers cancer diagnostics, donor screening. So you can see that there are different applications that can be done under the umbrella of human genetic testing. And one of the key advantages we can convince these customers are the high molecular weight DNA, as I showed you, the high integrity, as well as the consistent yields and purities we're getting, and the fact that we can customize kits based on their workflows. Of course, the high versatility with the sample volume and throughput is also a key argument. And the fact that we have bulk reagents, it's more affordable than compared to, for example, cartridge reagents when you're doing high throughput extraction. We also, as I mentioned, have CE IVD certified kits, which is suitable for labs that have to fulfill some regulatory requirements. We are very established in biobanking. So I was also recently in the Europe Biobank Week conference where we gave a talk and we showcased our involvement in the NARCO study, the largest German population study, uh, which was done at the Helmholtz Munich Center. So we were enabling the extraction of more than 200,000 Buffy coat samples, and they were satisfied with the yields. 99.5% of the samples were achieving the required yields, and they were seeing good downstream success in sequencing as well. We've also been involved in the Mayo Clinic, all of us population study, where they've really tested our technology with all kinds of different downstream assays, and it's been shown to really be robust. We were also involved in the UK 100,000 Genomes Project, and also another one from Decode Genetics, the one here you see in Iceland, which was the largest long read sequencing project where they did extractions with the chemagen 360, and they were able to put 3,000 over 3,000 samples directly into Oxford Nanopore long read sequencing. So there are publications, there are citations, they're all available in our brochures, and these are good convincing arguments if you have similar customer profiles. So the fact that also we can meet automation requirements is also a good argument here, especially when they need to process a large amount of samples. This is the donor screening customer. They're called DKMS Life Science Lab. They're based in Dresden in Germany. And if you're interested, we have user meeting, a chemagen user meeting that will be jointly hosted by them on the 25th of September in Dresden. So if you're interested to take a look at their labs, there'll be a lab tour provided. You can also invite customers to come and join us. This is a great networking opportunity to meet other users in the region. We have customers presenting talks on their applications. So this will be a nice networking event, which we'll provide you more detail on later. But this lab is really unique in the sense they have millions of samples to analyze per year. They're analyzing mostly swabs or blood, and they have developed a novel sort of high throughput HLA typing based on Illumina NGS, where they have pooling samples and then deconvoluting them. So they are using here in the picture you see the chemagic MSM1, which is the first instrument before the 360. It has the plates arranged in a long linear fashion as opposed to the turntable. But many customers, you may be surprised, there's still quite a few in the field. Many customers like this setup as well. You can place all the plates at one go, and they're still using this, and they're also upgrading potentially to different rod head systems. So they're really a high throughput lab, and they like the robustness of the chemagic separation technology. They've been using our instruments for over 10 years now and remain very happy. And there's a testimonial in our brochure about them. So you can read more about them on their website as well as this publication here. We also have medical genetic laboratories in hospitals or in universities. So this is actually from the Erasmus MC University in Rotterdam. They were using our chemagic system for large volumes, so 5 mils of blood, and they liked the real nice reproducibility they saw with the two different replicates here you see in red and blue. And they have many different diverse assays downstream, including MLPA or gel electrophoresis, long-range PCR, and sequencing. So the fact that they can get a large amount of genetic material to be able to cater to all these different assays is an advantage. Not many other platforms can extract from such large volumes. They usually have to pool two samples or multiple samples to meet this requirement. But with our technology, we can extract directly from 5 mils of blood. And yeah, with a high reproducibility and consistent results. We also see a growing demand for RNA extraction. We have validated different RNA collection tubes or blood collection tubes, such as the PAXgene blood tubes or the SASTET blood tubes, and we can achieve good RNA RIN scores, about 7 to 8. There are different applications for these, but we have various customers doing it for RNA-seq and so on. So there are application notes available as well. We see also dry blood spots coming up not more and more. The advantage being that they're easy to transport. You don't need a large volume of blood. They are especially key for newborn screening or newborn testing, where you can just get a small amount of blood from a newborn or even children. So these are usually used in genetic testing labs. We and also sometimes for infectious disease testing, but mostly in genetic testing. So with the fact that Revity also supplies um, the whole workflow. So from the punching of the cards, we sell the paper as well. We sell the puncher. We offer the extraction capability. You can do this on the chemagic 360 or the Pepito or the Prime. On the chemagic 360, we now have the ability to fully automate it. So we don't have to remove the punch anymore. You can process it directly on the instrument. This is something quite new, which we're beta testing at the moment, but it's looking positive. Following the extraction, you can do QC again with our lab chip or Victor Nevo for quantitation, or you can put it directly into library prep and NGS, for example, with the BioQ if you're doing a lower throughput extraction and analysis. And these, the quality as well is good. As mentioned, it's suitable even for next generation sequencing. Another sample type we deal with is FFPE tissues. These are largely used in cancer or pathology labs where they're preserving the tissue in paraffin and formalin. These are challenging material because they tend to be more degraded, the DNA. They are fragmented. They are sometimes modified due to the cross-linking or the formalin. So it's important to note that the expectations here are also manageable. It's really based on sample. We offer the chemagic extraction kit. Customers have to do an upfront treatment to remove paraffin, which can either be done with organic solvents or a high heating step, which we recommend, or this ultrasonic disruption by covariates. We're not really seeing much of this ultrasonic disruption so much. We are more or less focused on recommending the high heating step. It's the easiest to implement. You have also customers asking for RNA and DNA from FFPE, and it's important to note here that by incorporating DNase or RNase digestion after the heating step, you can essentially extract from the same sample, DNA and RNA. You just have to sort of split the sample, treat it with RNase or DNase, and then put the full the final solution onto the chemagic 360. So the chemagic 360 can then do the extraction, be it of DNA or RNA in the same run. This is again time saving and cost saving. It doesn't need cartridges. So essentially, and it also preserves the integrity of DNA, as you noticed. So depending on the initial sample material, of course, you can get quite good extraction quality here. Another target or material which you are seeing more and more is cfDNA from liquid biopsies. These are challenging targets because they're very low in abundance. There are about 170 base pairs in sizes, so they're small as well. Sometimes they can be fragmented. This is the only kit where we also sell a manual or recommend a manual extraction kit because the throughputs we know may not be high enough to justify an automated extraction. Hence, we say if you want to test our technology, you can test it. with this manual kit. We've shown that it works comparably to the chemagen column kits. You can get similar yields, and you can scale this up in when you're doing higher sample numbers. So on the machines, it's the same buffer chemistry with the same beads. So you can essentially scale your extraction. And this has been the fact that we can go up to large volumes with the chemagic 360 is also an advantage here. You can see up to here we have a linear increase in the cfDNA we've obtained. We have a cfDNA assay on the lab chip as well. We can co-mark it. You can see that this is a typical profile on the lab chip. The main peak is here. cfDNA are wrapped around histones, so if it's around 1 histone, it's 170. It's around 2 histones, you see it around 300, 3 histones, so on. The only issue, sometimes you might see genomic DNA contamination, which you will see sometimes as a peak around here, which is largely due to the plasma preparation. So it's really important when you're talking to these customers, asking them how they're preparing their plasma. There are double centrifugation steps required to remove leukocytes before you do the extraction, as the key thing here is to really extract the circulating DNA and not DNA in cells. So this is a growing field. We see that the demand is increasing, and we have really good reference customers for this. We have Professor Niels Pallesgaard in Denmark, who is using the chemagic 360. He's done a webinar for us, showcasing a comparison of the chemagic system with different kinds of extraction technologies, and ours was really well performing with the kind of different QC assays he runs. He did digital PCR downstream. So we promoted the webinar with Bio-Rad, which got really good responses. We are also looking at another cfDNA webinar potentially end of this year, so I'll let you know about that. We have other applications, for example, fetal RHD, rhesus factor typing, aside from cancer. So from cfDNA, you can also see cfDNA from the fetus in the mother's blood, and people are using this to type rhesus factor. So our extraction kits have been used for this application as well. And yes, we have also another interesting company in Biopsense potentially using our cfDNA extraction also in their new product development. So these are the different instruments you can apply to the whole workflow. And as well as cfDNA, we have microRNA or cfRNA. We noticed that our microRNA kits, although catered to smaller fragments, microRNA can work as well for longer RNA. So if you have customers wanting to also extract circulating free RNA, this is the kit to recommend. We've done a couple of app notes on this. It can extract good diversity of microRNA at comparable yields. So really fast as well. This is only available in an automated version, so on the 360 for sample volumes about 200 or 1 mil. And for both cfDNA and microRNA kits, they're usually working with clear body fluids like plasma, serum, or exosomes as well. Urine as well has been done. Yep, so this is already what I showed you before. Basically, again, with all our kits, it's catered towards getting longest possible length fragments. So you can already say that our extraction technologies have been used for long read sequencing. There's been a host of different publications to demonstrate this. This one being the one from Decode, which is, I think, the most convincing one. This is infectious. As mentioned, COVID really increased our footprint in this area, and since then we've been also being able to convert the labs past this COVID detection phase into pivoting their technologies to detect different pathogens like HPV, for example. But of course, there's a lot of work still to be left. There are a lot of instruments still sitting in basements, not being used because of the drop in demand. So really the goal here is to convince customers to use the instruments for different extraction workflows, be it even in human genetic testing or in different sample testing, or if they're into wastewater, we have done that as well. So yeah, for parasite pathogen screening, we have a really big customer in Brazil. They're called Fiocruz. They're a nonprofit, but they have a arm that manufactures vaccines and also diagnostic tests. So they have developed this diagnostic test called KitNet Plus. They've run it with our extraction chemistry on the primes. They have a large number of primes, and they are doing 552 blood bags a time at a day. So it's really an impressive throughput. We've highlighted them in the Prime user stories, and they're really doing a lot of blood screening for the Brazilian population. And this helps in making sure that the blood supply is suitable for donations and reduces the waiting time for anyone who's been to an endemic area to be able to donate blood again. So they're really doing this at high throughput. Uh, we've also worked with the Red Cross, and we've able to get good detection sensitivities of different viruses. And the key advantage here is the fact that we can use our extraction platform with large sample input volume. So because infectious disease labs, they have this need also for high throughput. But we have the ability to pull samples as well with the 96 rod head, so you can even go up to 1.5 mils, sometimes even 3 mils if you want to pull more samples. So this is something that customers can do to increase the detection of their downstream viruses. And we've also our fully automated instruments like the chemagen Prime Junior is also a very good one that's been used in Spain for this application. Another nice thing to highlight, we had recently a blog post on automation for labs looking to employ automation is the advantages of using chemagic extraction. The fact that this lab had to handle so many different sample types with 23 variations of 5 extractions because they had samples not only from humans but also from pets. Um, but with the chemagic technology, they could really consolidate their workflows down to one extraction with two upfront variations, so pre-treatment variations, a hard lysis and soft lysis based on the different pathogen they were dealing with. And there they could really reduce the cost in terms of reaction or also in terms of how many people needed to run um, this workflow and increase their throughput as well and reduce their repeat rates and error rates. So this is a real success story. I feel that we should also highlight to customers. As mentioned, with SARS, we were able to meet the real high testing demand there, and I'm hoping there's no other pandemic, but if there was, our instruments are really good for this. And we've seen now with wastewater surveillance, this is something that they're doing in US and Europe, where they're looking at the cycles of viral particles in sewage or wastewater. You can't really link this to the um, the incidence of flu or incidence of COVID in the population. We have two publications where we have two different kinds of workflows that can handle wastewater. You can either do it directly from wastewater, involving some centrifugation steps up front, but lysis is done and then you add the whole thing on the 360. You can do it up to 10 mils, but we've also developed now even up to 20 mils. So this is something you can also tell your customers. Alternatively, you can run the wastewater through a filter and then um, homogenize that filter with beads, with lysis buffer, and this lysate can then be also done on the extraction instrument at higher throughputs due to the lower volume. So this is also a workflow that has been done. Okay, I shouldn't go into too much other detail. So there are other kind of niche applications that involve plasmid isolation or soil, but I would like to sort of stop here to take questions. So,
Yeah, thank you, Catherine. We have some questions here.
okay.
Uh, so the first question is, just a minute. Uh, is the DNA consistency, the yield, is consistent throughout different samples? Is it consistent with different starting materials like the blood or plasma, or does it vary usually?
Yeah, so it really depends on the sample source as well. With blood, there can be the blood may be from patients or from healthy donors, and we've seen that with from patients, for example, suffering from leukemia, they may have a higher white blood cell count, and in that case, their yield of DNA is also very high. So it really depends on the patient if they're suffering from any disease. Some yields may vary, but with the healthy donors, you'd see a typical consistent yield of between 20 to 50 micrograms per mil, and this is in line with the white blood cell count that they have. And with other sample materials like saliva or swabs, these may be a bit more variable because of the form of collection, but I've also seen in large saliva population studies, you can get quite a consistent range of about 60 micrograms as well, depending, yeah, on the sample input.
All right, thank you. Other questions about the beads. I mean, do we sell these beads separately? The
Yes.
chemagic beads, for example, for the NGS workflow, like example, like for what we use for the ampere beads, or does it
Mm.
come all in the kit size?
Yes, we do sell the beads separately. So if you have a specific request or specific opportunity, just let us know. We are developing cleanup beads as well, similar to the Ampure. I think these are still a work in progress, a product development at this stage. But we do sell our beads functionalized with different groups with different applications. And I'm not the person in charge of this product. I have a colleague who does this, who has more insight. So if you have a specific customer with a request, just let us know.
Great. What usually is the DNA yield for 1 ml of cfDNA or liquid biopsies?
Yeah,
Usually, do we have a
yeah.
range
This
for
is
them?
yes. There is a range and it's very low. So it's between 1 to 30 nanograms per mil. I think for cfDNA. So or 1 to 30, yeah, 1 to 30 nanograms 1 to 30 nanograms per mil of plasma. So it's really variable as well. For cfDNA, you because it's such a low abundant target, um, it's in terms of quantifying cfDNA, there are different methods, and you have to also be aware that there are different biases towards each method. So most people would measure with qubit, which not only measures your cfDNA but also other nucleotides present in the alloid. And if you're using a kit with that uses a carrier like poly-A RNA, for example, this contributes to your qubit results. So that's also something to note. We've noticed that the QIAGEN kits they lead to a higher background when you're measuring with qubit because of their use of poly-A RNA. We've also had to use poly-A RNA just to make sure we don't that we're not losing out to them in terms of this readout. But this is something to note. So only in tape station or lab chip you can see the peak, and there are methods to quantify this. Uh, this can also be variable if you're using an assay not dedicated to cfDNA. So you need a marker that has to migrate around 50 base pairs to be able to quantify this peak accurately. If you're using an assay that is not meant for cfDNA, these markers can vary based on the different extraction kits. Then you have a really unreliable means of quantifying your cfDNA. How we quantify is also with PCR, with this allo repeat region that you find on cfDNA. Um, but yeah, it's there. You also it can be subject to variabilities, but this is what we've used as well. But
Thank
yeah,
you.
it's a typical range of 1 to 30, let's say.
Two questions on the schematic version, schematic 360 version. Does the CIVD version of the instrument have any limitations compared to the RU version, or
N-
is the
No,
functionality the same?
they're identical. I mean, I think the IVD version comes with all the documentation. They're all shipped from Turku, so the IVD version comes with the documentation to meet the regulatory requirements. And yeah, it's just to comply with the IVD needs. So if you buy the IVD instrument and you use an IVD kit, then you're compliant with the the IVD regulations. But essentially, the technology itself is the same for the RUO. Yeah.
Okay, a follow-up question on that is can the roads be changed by the customer, or does it require a service professional?
No, it requires a service engineer.
All right. Um, One few questions on the viral nucleic acid extraction. Can we use whole blood on viral nucleic acid extraction kit?
Say that again?
Can we use the whole blood
Oh,
for
for
viral?
yes. Yes, there are kits that can process that are suited for blood as well. So I think it's we have the IVD 1049 kit that can be used for blood samples. So it includes a blood lysis buffer. And we have a comparable IO kit as well.
Okay, and
Sorry.
we are also looking for a universal kit for viral extraction. Is there or does it depend on some gram negative? Does it extract all the virals and bacterial, or is it like for specific for gram positive or negative?
No, the gram positive and negative is really dependent on your upstream processing. So gram positive is harder to lyse. So you might see that you might need um, more stringent lysis conditions, and this can be done with heating usually. But if it's a, let's say, plasma sample, you can use the same kit for different kinds of bacterial samples or viral samples. The only thing is if you're using like stool as a sample material, then you might require a stool lysis buffer to be able to get these different pathogens. But I would ask you to always write into us and let us know what exactly is the sample material or the pathogens, and we can provide you a recommendation.
Okay, thank you. Question on the wastewater. Has the wastewater samples been validated on other pathogens beside COVID, or do we
Yes,
have any app notes?
yes, it's been done for influenza, RSV. The app notes that we have, I don't know, I don't think they mentioned this, but we have done it before with different viruses, so it's not just COVID.
Okay, I think there's one question on the bioanalyzer, or I think you already answered it on the CFDNA.
Mm-hmm.
Do you recommend relying on the bioanalyzer to quantify CFDNA, or it should be through PCR?
Mm. Yeah, as I mentioned with the bioanalyzer, it's if you have a marker, an internal control marker that migrates at 50 BP, that could be a reliable quantification. Um, but if not, then it is subject to variabilities. And with qPCR, that is what we use. So we do use this to quantify the cfDNA.
Awesome.
I mean, digital PCR is also something that's more sensitive, which you can use.
Okay. And I believe we have an app note on using this. We're using Bio-Rad, right?
Yes,
Yeah.
we have a case
Yeah.
study on this. Yeah.
Yeah. Perfect. Uh, that's all the questions we have on the. Is there any? So we have 2 minutes. I give the audience a chance to raise their question. If you have, you can raise your hand and feel free to talk, or feel to type in the chat. We just have a minute or so.
Oh, what about the poll? Can I see the poll? Ah, yeah.
Yes. Yeah.
Okay. Okay, 81% diagnostic labs, human genetics, 22% biobanks, 70% diagnostic labs, infectious disease, 15% pharmacy. Oh, okay, that's really nice. So that's a almost even split between human and infectious, more in human,
Mhm.
and a smaller footprint in biobanks. Okay, thank you for that.
Yeah, one last question on the FFPA before we wrap up.
Mm-hmm.
The You mentioned about the high heating step compared to the xylene or the organic
Yes.
solvent method. Is it, do we, using the same number of rolls, do we expect more concentration of the DNA or RNA compared to organic, or do we have some kind of a tech note for that?
You've been comparing the different pre-treatment.
Yes, yes.
We don't have an app note for that, like comparing the different pre-treatment. Um, and I think it really depends on the lab as well. Sometimes they are used to a certain technique. So, for example, we have a big customer in the US, and they're using xylene, and they're used to that, even though they're doing a high throughput. But depending on the lab may not want to work with organic solvents, then they would prefer the melting technique. So it's really also what the lab prefers as regards to with the yield. I don't know if that makes a big impact on the yield that comes out of it. It's really, I think,
And
implementation. Yeah.
and of course the heat treatment is much more easier compared to
Yes,
working.
exactly. Yeah. And we found that this
Oh,
doesn't also impact RNA so much. So that's been tested.
great.
Hmm.
All right, that's about it. Thank you, Catherine. Thank you for the enlightening presentation. I really appreciate it. And thank you for
Thank
your
you. No problem.
questions. Yeah,
Yes,
thank you
thank
for your
you.
questions. And feel free to write to us if you have any questions, and we'll get back to you on that. Thank you.
Thank you. Bye.
Bye bye.